Good evening, everyone. I'm Alan Fisher. On this episode of AZ Illustrated Science, some plants can look alike but belong to different species. Telling them apart may require studying their genes. We'll have that and talk with a researcher about a new technology that could help people to see. But first, here's a look at today's top stories.
A countdown clock is ticking down for the next University of Arizona-led space mission. The clock on the UA campus is slowly ticking down from 999 days to an early September 2016 launch of the OSIRIS-REx mission. The goal of the project is to send a craft to an asteroid, gather samples, and then come back to Earth. The timing of the launch is precise.
The asteroid is not going to wait for us. We have to get off the surface of the Earth starting in September of 2016.
The mission is in the testing phase right now. If all goes well, the construction of the craft will begin in earnest early next year. The Tucson area housing market continues to rebound, with selling prices up 10% from a year ago. That's according to the Tucson Association of Realtors. That group says the median sales price in November was $159,000, up nearly $15,000 from a year ago. The association also reports that more houses are going on the market, nearly 5,300 in November, up 19% from a year ago. And the Community Food Bank of Southern Arizona is turning to the public for some financial help. 4 local families have pledged a dollar-for-dollar match up to $100,000 for donations made during this year. And that's a look at tonight's headlines.
As we have discussed many times on this program, genetic research is opening new areas of exploration for scientists. Heather Gray tells us how researchers are using genes to determine taxonomy. You know, what belongs to what species, genus, family, and so on. In some cases, scientists are even reevaluating such classifications.
For local plant breeder Mark Dimmitt, DNA holds the answer to a compelling question. The plants in his collection look very different from each other, and their native habitats are spread out across an enormous continent. So how are they related?
Mark is an expert in his field of botany in general, but adenium specifically, and he has great samples of individuals from all over Africa.
The University of Arizona Genetics Corps is a lab that helps researchers, scientists, and even members of the community use DNA to answer their questions. Dimmitt contracted the lab to extract, sequence, and analyze DNA from some of his wild specimens.
Yeah, bury that right there, just like that. That's good. I was the first or one of the first people to hybridize adeniums.
Adeniums are a genus of flowering succulent. They are native to Africa and live in isolated populations from South Africa to the southern edge of the Arabian Peninsula. Dimmitt brought them into cultivation from the wild as an ornamental more than 30 years ago. He began selectively breeding them for traits like brighter and bigger flowers.
That's probably good. It's the only succulent plant that has both a beautiful sculptural form and puts on a spectacular flower show over a long season. I don't know any other plant that you can say that about. The taxonomy of the genus Adenium is uncertain. The biggest ambiguity is the complex of adeniums that occur in a band across Africa south of the Saharan Desert. They range from tiny dwarfs only a foot high to 15-foot high trees. And they've been named as 3 different species. But it appears that they integrate, at least in cultivation, they definitely do, and maybe they do in the wild, in which case they may be one very variable species. So I hope to answer that question with a genetic analysis.
For me, it's his central question was just, hey, these plants from these different areas look very differently. So looking at just their shapes and forms and such, there are clear geographic divisions. Now, do geographic divisions equal species?
Genetic analysis is becoming increasingly important to scientists. because looks can be deceiving. Dimmitt says two plants from the same species living in different climates can look very different, or two separate species in the same habitat could look similar because plants adapt to their environment. Dimmitt says taxonomy based on visuals alone has its limits. Kaplan says DNA is a powerful tool.
He has a world-class collection of Adenium plants, and he was able to bring in just a single leaf from each one, and that was much more DNA than we possibly needed. And so we ground them up and basically washed away everything but the DNA. We amplified out 5 different parts of the Adenium genome. With those different parts represented, we're going to get a fairly good idea of what's going on with the relationships of all these different populations of Adenium. Oh, that's right. Yeah. So there were mitochondria, chloroplast, and nuclear. What you're always striving for in this kind of work is the species tree. So this is the evolutionary history of these organisms.
The analysis will yield a map of the plant's family tree, or a phylogenetic reconstruction. Mark Dimmitt has spent 3 decades trying to understand those relationships through observation alone.
I was given the data, and then I ran it through different really specialized phylogenetic reconstruction software to get a bunch of tree diagrams. And Mark and I are going to sit down and go through that all and see what it looks like.
The quantity of information coming out from genetic analysis is mind-boggling. And it's also causing huge changes in taxonomy because we're finding out that some of the things, plants that look alike, animals that look alike, really aren't that related at all, so we have to change their taxonomy in that case.
To Kaplan, the trees are meaningless without the knowledge and interpretation Dimmitt provides.
These were ready, they have beautiful root systems.
That is the magic part. Really, what Mark brings is really unique in terms of the expertise, the knowledge of what these plants are and how they live their lives.
Tomorrow, an FDA advisory committee will review a new allergy pill. This pill is not an antihistamine or steroid. Rather, it works like an allergy shot. The pill actually gives the patient a small dose of allergen to teach the immune system to tolerate it. Such immunotherapy pills are already available in Europe and mark a possible new treatment approach for severe allergies in this country. Recently, Anna Augustowska sat down with Dr. Tara Carr to discuss one potentially very dangerous form of allergy, food allergy, and possible new treatments like the immunotherapy pill on the horizon.
Good evening, doctor. Thank you very much for coming in today.
It's my pleasure to be here. Thank you.
Food allergies are often confused with food intolerance. So please explain for us what a food allergy is.
That's a great question. When many patients use the term food allergy, they use it to describe the symptoms of I eat a food and it makes me not feel well. But when doctors use the term food allergy, we try to be a little more specific and really use it only for those patients who have a specific abnormal reaction of their immune system when they're exposed to certain foods. For many patients, this is manifested as what's called a type 1 or immediate type allergy, which is that classic anaphylaxis picture that we see in patients. But there are other types of food allergy that can develop as well.
What are the most common allergens?
The most common allergens are milk, egg, wheat, soy, peanuts, tree nuts, fish, and shellfish. Together, these account for approximately 90% of the severe or immediate type food allergy reactions.
Could you tell us more about this life-threatening form of food allergy, anaphylaxis?
Yeah, absolutely. So anaphylaxis occurs when somebody ingests something that they're allergic to, and a specific immune reaction happens where cells called mast cells and basophils release chemicals that cause allergic symptoms. And if it's to a degree that's severe enough, it can be life-threatening for patients. So the most common symptoms that patients get when they're suffering from anaphylaxis are skin symptoms, so itching, flushing, hives, like get big, red, itchy bumps, swelling of the eyes, swelling of the lips, swelling of the ears, the hands, and the feet. Sometimes patients have involvement of their respiratory system, meaning coughing, wheezing, chest tightness, chest heaviness. Sometimes their eyes will get red, sometimes the nose will get stuffy. Gastrointestinal symptoms can include feeling nauseous, vomiting, really bad diarrhea, belly cramping, and some patients get low blood pressure and some symptoms associated with that, like feeling lightheaded or passing out and losing consciousness. And all taken together, these symptoms can be life-threatening if they go untreated.
According to FIRST research, food allergy research, now we have up to 15 million Americans suffering from food allergy, and this number is growing. Why?
That's a great question. The allergy in general is increasing in our society. Food allergies as well as other types of allergy, allergic rhinitis, nasal allergy symptoms, asthma, skin allergies. So part of it might be that the interaction that we're having with the environment is different. I don't know if we can say that the people are so different than we were 50 years ago. So it might be a question of exposures, different exposures to the environment, or it might be a different way that our bodies are responding to those exposures.
Is there a way we can regain tolerance to the food since we are losing it, obviously?
That's a good question. Up until now, the main treatment for food allergy is really avoidance of the foods that you're allergic to. The food allergy specialists and researchers and scientists across the world have been looking into this very aggressively and are trying to develop ways to reverse the immune reaction when you're exposed to foods. The two different mechanisms that have been most widely used so far are called sublingual and oral immunotherapy, meaning either putting the food underneath the tongue or eating the food. Now this is done in a very controlled environment under the care of doctors and very specialized nurses and research personnel, and in a very controlled way. And over time we're trying to have people be able to tolerate the different foods that they might be allergic to. Most specifically and commonly looked at are milk, egg, and peanut. These certainly are not treatments or therapies that people should try to do on their own at home. They're still in the early stages of research, and so we can't say this is ready for prime time or going to be available at your pharmacy or at your doctor's office, but we're certainly hopeful as a medical community that some of these treatments will really help. A newer type of immunotherapy trying to reverse food allergy is called epicutaneous. So on the skin, basically applying patches that contain some of the ingredients that people are allergic to, specifically peanut, and seeing if that can reverse what people, how people respond when they're exposed to these foods. Again, that's also not available to patients yet, but certainly is interesting.
How we as a community can protect children from food allergies, since it's a big issue with children, how we can protect children?
Yeah, that's a really good question. Because we don't have a way currently to reverse food allergy in these children, or even really to prevent food allergy from developing, it's important to know how to prevent a food allergy reaction and then to treat the reaction when it does happen. The prevention, of course, being making sure the children are not exposed to those things that they're allergic to. So this is something that parents have to become very involved with, as well as other caregivers, schools, and the communities, the organizations, and the various activities that the children participate in. There are great guidelines available on the internet through either the Arizona State resources or some of our national allergy resources for how to go about identifying your child as food allergic, wearing ID bracelets, coming up with action plans that are written, that are signed by a doctor, that have medications attached to them so patients are aware of how to treat symptoms.
We have about 30 minutes left. Could you please tell us how does the dynamic within the family changes when you have an individual suffering from food allergy?
That's a great question. My nephew was recently diagnosed with peanut allergy, and I can tell you from firsthand experience, it's very difficult for families to find foods that are safe for their children or for their loved ones to consume. It's often a combination between of all the family members, and it's been shown to actually be more expensive for patients to try to find foods that they're not contaminated with their allergens.
Thank you very much for coming in today.
You're very welcome. Nice to meet you.
I'm Judy Woodruff. On the next NewsHour, world leaders gather in South Africa for Nelson Mandela's memorial. That's Tuesday on the PBS NewsHour.
The Obama administration says the plan to temporarily loosen restrictions on Iran won't end sanctions.
Any oil company that thinks now is the time to get back into Iran will find out that they are on the wrong side of the sanctions. Likewise, any bank.
I'm Renee Montaigne. How much Iran benefits from the temporary deal and its nuclear program on the next morning edition from NPR News.
A handful of companies around the world are working on retinal implants designed to help people with degenerative conditions recover their vision. Such implants, however, only allow people to see in low resolution. A local researcher is working on a new form of retinal technology that he says could help patients see in much greater detail. That researcher, Wolfgang Fink, is here with me now to explain. Wolfgang, thanks again for joining us again.
Thank you for having me.
Wolfgang, could you briefly explain what retinal implants are and how they work, please?
Yes, retinal implants are devices which allow a blind person to attain some basic vision back. They usually consist of some eyeglasses which the blind person would wear. On top of the eyeglasses is a small miniaturized camera mounted which sends wirelessly images to an external processing unit. The external processing unit subsequently reduces the resolution of the image and does some extra image processing and sends the images inside the eye to a chip which then subsequently electrically stimulates the retina to create some basic vision.
And Wolfgang, if I were suffering, say, from macular degeneration, at this time and with the current technology, what would I be able to see?
Well, with the current technology, what people or patients tend to see is some basic blurry shapes. They can distinguish light from dark, and they may also be able to sort of localize where certain objects are and also count them. However, it takes a huge effort still to sort of scan large-scale fonts. So if you have a large letter, it takes about several seconds for a subject to scan the area to then determine what letter it is. We have here a little simulation which shows a 10x6 implant, which is the resolution by the currently only FDA-approved implant, the Argus 2.
So this, Wolfgang, would be the best available technology in the US?
At this point, yes. So this shows a 10x6 resolution image. So as you can see, you can make out some motion going past the camera, which is built into the screen of the computer. We can now improve this by adding some contrast to it and also potential other image processing filters to smoothen out the image or to help the patient, for example, make out a door or a window. Now, if we go further, for example, in Germany, there are implants which have on the order of 1,000 or 1,500 electrodes. So I'm dialing in now a 32x32 implant. So as you can see now, in principle, if the stimulation was done right, which it isn't at the current time, you would be able to potentially make out faces. You could also see that now the object moving by is actually a hand. We could now, again with image processing, change the appearance of the image for the blind.
And Wolfgang, how will your new technology improve people's ability to see?
Yes, so our technology is actually geared towards how to control existing implants, both in the US and in Germany and elsewhere. Also, I should mention the Australian bionic vision implant, who we collaborate with. It's a suprachoroidal implant. The idea is, think of you having a printer at home and you download a new driver software which gives the printer new capabilities which it didn't have before. That's what we're doing. We have new stimulation paradigms of how to communicate the processed images as best as possible via the electrode array to the blind person.
Okay. And you mentioned stimulation. How does this vary versus increasing the number of electrodes versus the stimulation you mentioned?
As I showed in the simulation, while it's a very good goal and should be done to increase the number of electrodes on the same area, which means the density of electrodes, If this does not go along with a change in how to stimulate and actuate these electrodes, it will not do a thing. It will basically just wash out any subtle detail because of a physics effect called crosstalk. Now, in our case, our simulation paradigm will afford a blind person to take advantage of the potential high resolution which the chip can deliver based on the number of electrodes. So in other words, what we are trying to do is to have a one-to-one correspondence, more or less, between one electrode and a image-giving pixel. So in other words, if you have 1,000 electrodes on your chip, you would like to have more or less 1,000 discrete pixels.
And Wolfgang, where do we stand in terms of commercializing this? Is this something that's going to come out of the technology transfer department at the university?
Yes, Tech Launch Arizona, as a matter of fact, has filed a patent on the new stimulation paradigm. I should also at this point mention my colleague in Germany, Dr. Erich Schmidt from the University of Tübingen. We have been collaborating on this project for several years now, and he's also a joint inventor.
Okay. And in terms of this type of technology, does this have any other medical applications, Wolfgang, in terms of perhaps neural stimulation, that kind of thing?
Very much so, as a matter of fact. The stimulation paradigm could be applied to deep brain stimulation, could be applied also to paralysis, if you have electrodes to reactivate limbs which are paralyzed because the muscles are still in place. However, there's just a disconnect from the brain to actuating the muscles. And so with this stimulation paradigm, we might be able to get more refined control over the muscles.
Okay. And briefly, Wolfgang, we have about 30 seconds left. When can we expect to see the increased stimulation of retinal implants hit the marketplace for people who are needing this device?
Well, that depends actually also on our colleagues, whether they are willing and able to work with us to implement the current stimulation technology into their existing implants and future implants. Now, the Australians, at this particular point, are working with us to make this happen. And technically, it is possible to implement some of those things as of right now.
Well, Wolfgang, thanks so much for joining us again today.
Thank you very much.
In our next interview, we discuss the emerging field of translational Bio-Informatics. Joining me to explain is Dr. Yves Lussier, a professor of medicine and the new Associate Director of Informatics for the Bio5 Research Institute. Yves, thanks so much for joining us today.
Thank you for having me as well.
Our pleasure. Yves, could you explain translational bioinformatics and what that is?
Yes, it sounds like a mouthful, but since the advent of the human genome in 2001, there's this nascent field of trying to translate in words that physicians could understand the meaning of the genome for clinical practice. And that's the role of translational bioinformatics, using computers and mathematical analytical tools and a bit of artificial intelligence as well.
And Yves, how is that helping physicians improve the way that they treat patients? Could you give us some examples, please?
Certainly. I think the most eloquent examples are the work of Atul Butte from Stanford and myself. We have repositioned drugs and that are currently tested in clinical trials using first computers. So in high throughput, we use massive computing facilities and a good model of the cells in cancer, and figured out new use of drugs that were not understood as having a role in cancer. Thereafter verified in cellular and animal models in less than 6 months and confirmed that our hypotheses were correct. And we were in clinical trials within less than 4 months, in my case with some NIH funding, which means that from concept to clinical trial, it took 2 years. If you do it the traditional biological way, it could be as long as 6 or 17 years.
So it's much quicker and kind of customizable for the patient then?
And in addition, that's right. We are finding patterns that are unique to some individuals and which would require certain unique approaches to their treatment that are unconventional and would not proceed with what would be a boilerplate or an average treatment of that patient. Possibly taking drugs from a different cancer to treat them would be more appropriate, because the mechanisms that are at play are more analogous to that of a different cancer that occurs. We define currently cancers by their organ of origins and cell type of origins rather than by their molecular mechanisms that are at play. In other words, to repair the motor of a car, if someone had switched the motor from a different company's, let's say a certain company's car for a different one, a Japanese company's car into an American car, you'd probably take the approach of taking the parts to replace the motor that come from the Japanese company, even though the appearance of the car is an American car.
Okay. And is what you're studying, does it relate to the recent case we read about in the media with Angelina Jolie having certain markers, predisposition for breast cancer, and had surgery as a result? Is that kind of related to what you're doing also?
Yes. So that's a case of the use of the genome as well. Single gene diseases such as Mendelian disease, we know of ataxia, Friedreich, and other rare disease, cystic fibrosis, another common one. So some cancers are also Mendelian disease. They're caused by a single gene, and those are the very rare cases. BRCA1, BRCA2 genes in breast cancer increase the risk up to 50% in some women to have this breast cancer, and at very high levels of ovarian cancer. And so this approach of treating patients using genetic information, single gene, is more conventional, and that's what traditional genetics has brought to genomic science. And now geneticists as well as translational bioinformaticians are working together to understand the complexity of the interactions of these genes and why in some cases it's really a larger number that we need to understand altogether to provide the proper treatment. Generally, it's a sufficient number to recognize the molecular pathway. An analogous would be to say, if I name the houses and describe the houses, can you recognize the street I'm speaking of? And that's the pathway. So right now we're just giving street numbers and house names when we're treating with a single gene like that, and in some cases that's the right choice. But with 10 years of experience since the genome, we know that the majority of diseases are called complex inheritance. They just are not well addressed by this approach, and we need to embrace this complexity and yet simplify it. So our approach is to try to recognize the streets from naming the houses on it first, and then bring a simpler language to physicians, which are the deregulated pathway, or in other words, for a cab driver, it would be the intersections of two streets, rather than naming each house on the street.
And Yves, we have about 30 seconds left. Could you tell me about the multiple roles you'll be playing here at the university?
So in addition to Bio5, I have a role as an Associate Vice President of Health Sciences in the Office of the Senior Vice President, where I will help create services from the molecular level up to the clinical level in managing big data and transforming data for improvement of clinical science and clinical care.
Great. Well, Yves, I want to thank you very much for joining us here today.
Thank you very much as well for having me.
That's our show. To post a comment on any of these stories or to keep up with the latest news, go online to our website at azpm.org. I'm Alan Fisher. Thanks for watching.